So with that, I will leave it to Tom to present some of the bacteria inhibition studies that we've been working on. As I mentioned, we were able to do some great work with him over the past few months, and I'll leave it to Tom.
Thanks, Jim. Yeah, if you could hand over the sharing of the screen.
Sure.
Thank you. And that was a wonderful introduction for PEKK that we have been studying and collaborating with Oxford for, I don't know, about a year now, really to look at bacteria properties. You heard a lot about great bone growth properties of PEKK, but I want to tell you a little bit about what we're finding in terms of bacteria inhibition. So just as a small background, in my career, nothing like breaking your 20-year career down to one slide, but we have spent a lot of time looking at nanostructured surfaces for orthopedics, for vascular applications, for antibacterial applications, really a wide range of medical device situations. And what we're finding, you can see on this slide some examples, but what we're finding is nanotextures are a great thing. If you can create them appropriately, you have a process that can create them, you have a material that can have nanostructured features. We're seeing properties like increased bone growth on 3D printed metals when we collaborate with companies for amputee type applications. Some images there in the upper right of this slide, we're seeing improved wound healing when we create nanostructured features on things like titanium abutments used for amputees. Even things like catheters at the very bottom, we're seeing antibacterial properties when you create appropriate nanoscale features. So we're seeing really a great ability to control cell function, whether it's decreasing bacteria, decreasing inflammation, or promoting tissue growth, just using nanotextures without drugs. But really, I want to concentrate on PEKK here and some of the exciting things that we're finding out. I think, as we all know, in the bacteria community, we have a real problem that is coming up. It actually is already present in terms of antibiotic-resistant bacteria. In fact, one of the specific examples that I like to give is a prediction from the Centers for Disease Control, which has predicted that more people will die from antibiotic-resistant bacteria than all cancers combined by 2050. So we are very interested in working with companies like Oxford who have a nanostructured surface to see if we can decrease bacteria function without using an antibiotic, without contributing to this problem that is down the road for antibiotic-resistant bacteria. So we did a number of in vitro results, both to see what the bacteria response is to PEKK, as well as to understand a mechanism of action. Obviously, we're not using antibiotics, so there must be a mechanism that we can explore for why PEKK is behaving the way it is. So following very standard colony-forming unit assays, we've looked at a number of different bacteria. We've looked at Staphylococcus epidermidis, which is shown here. This is the bacteria that's on your skin, easily infects wounds during orthopedic surgery. It comes with you to the surgical suite, so there's no way to remove it. And what we're seeing, obviously, is comparing to titanium or PEKK, that we have significantly decreased numbers of colony-forming units on PEKK. PEKK is here in the yellow, and this is just after 1 day. So a very quick culture time, we're seeing this significant decrease in bacteria colonization on PEKK. What is further intriguing to us is it doesn't seem to matter the type of bacteria that we're culturing on PEKK samples. So this is an example for Pseudomonas aeruginosa. We often call this pseudo. So Pseudomonas aeruginosa, same trend. In the yellow bars, again, that's PEKK. You can see a significantly decreased colonization after just 1 day compared to PEKK and titanium. And of course, the one that we love to study in my lab is MRSA. MRSA is methicillin-resistant Staphylococcus aureus. This is the antibiotic-resistant form of Staphylococcus aureus, so you cannot use the antibiotic methicillin to kill this bacteria if it colonizes your orthopedic implant or any medical device. And again, here we're seeing a significantly decreased colonization on PEKK compared to PEEK and titanium. Now, the other interesting thing is we have assays in the lab that can look at the number of live versus dead cells. It's another way to assess materials for antibacterial properties, same color scheme. And what we're seeing after the same period of time, whether we look at Staphylococcus epidermidis or any of these talking about, is a lower number of live cells. So even though I just showed you graphs of the decreased number of cells on the surface of PEKK, of those cells, a significantly lower number are alive. So it actually, makes for an interesting thing to study, is of those that are attaching, a significantly less amount are alive. In fact, 50% at some time points on PEKK, whereas we're seeing, close to 90% of Staphylococcus epidermidis alive after attaching onto titanium or PEEK. Same thing for Pseudomonas aeruginosa. Here we're seeing 50%, 60% of the bacteria alive. Again, significantly less numbers, but of those that attached, significantly less are alive on PEKK compared to titanium and PEEK. And the same thing for MRSA. When we stain for live dead assay, we're seeing significantly reduced number of MRSA that are alive on PEKK compared to PEEK or titanium. So quick summary before I go into a very brief discussion of why we think this is happening, is the results of our in vitro work have really shown decreased bacteria colonization for gram-positive or gram-negative or antibiotic-resistant bacteria on PEKK compared to PEEK and titanium. And of those smaller numbers that attached, less are alive. So we're seeing decreased number of living cells of those that attached on PEKK compared to titanium and PEEK. And impressively, if you were following close attention, you would see that MRSA actually showed the best inhibition on PEKK compared to the 3 bacteria that I showed with you. In other words, there were the least number of adherent MRSA compared to Staphylococcus and Pseudomonas aeruginosa. So let me briefly talk about why we think this is going on. And of course, you can ask why until you're as blue as the slide that I'm showing you. But here's some thoughts why we think PEKK is working the way that it is, improving bacteria inhibition. Many of you may know, if you start at the upper left part of this slide, that nanofeatures are all over nature. So nanofeatures are on leaves, they're on insect wings, they're really on a number of things that have evolved to control surface energy. So if you look at, again, something as simple as a cicada or a locust, and you look very high magnification at the wing of that insect, you will see nanofeatures, very discrete nanofeatures, discrete spacing at the nanometer level, discrete heights. Those features provide that insect with antibacterial properties. I might also add, we're finding out that they're antiviral properties too. So in light of this COVID-19 situation, we're asking ourselves the question, can these features also decrease virus adsorption and activation on medical device surfaces? But the other thing that we have done is we've modeled these interactions. So on the right, you can see that big red bacteria. This is your MRSA trying to attach to a surface that has nanofeatures. Our computational models really show that as the bacteria is trying to attach, it spreads its membrane so much that it either can't attach or these little nanofeatures poke the membrane. And that can lead to inhibition of attachment or death in some cases in our models. Now in our experiments, if you go to the middle of the slide, slightly to the left, you will see other samples. This is not PEKK. These are other samples in which we have created nanostructured features that are micron and nano structured. Now, if you look at the these are AFM images. If you look at the D micrograph, this is a picture or a description of how proteins adsorb to micron surfaces versus nano surfaces. And this protein in particular that we're studying is fibronectin. Fibronectin is a wonderful protein that can promote bone growth, promote bone cell attachment, and reduce bacteria attachment. So what we're seeing is when fibronectin absorbs to the micron surface, in D, you basically see these blobs that are on the surface. Those blobs are fibronectin, and it's absorbing in a non-bioactive state. But when we create nano features out of that same polymer, again, looking under nano, the letter D image, you see the fibronectin begins to spread, and it looks more fibrillar, more like fibers. Those fibers are more bioactive. So not only are we creating these features that can change how a bacteria membrane attaches or not attaches, but we're changing the energy of the surface. And changing the energy of the surface changes what proteins absorb and how they absorb. These proteins are nanostructured, so we're really interested in exploring these nano features to alter those protein interactions to reduce bacteria attachment. And you'll see at the very bottom, of course I don't have time for this, but we've actually created an equation. This equation can predict what size nanoscale feature you should put on your medical device to in turn give you the energy to manipulate these protein interactions that could reduce bacteria attachment. So real quick, that's what we did on the PEKK samples from Oxford, is we measured surface energy. You can see on this slide that compared to titanium, compared to PEKK, we actually created or looked at the surface energy of PEKK and found that it had closer surface energy to 3 key proteins in your body that are known to reduce bacteria colonization. So mucin is in your saliva, lubricin, casein, these are all natural proteins in your body fluids that have antibacterial properties. So what we found was that PEKK was able to have a surface energy similar to these proteins, and in turn, it enhanced the absorption of these antibacterial proteins naturally in your body. So here's some of the results. If you're familiar with ELISA assays, this is just one way to determine protein absorption on your surface. You can see PEKK had a 6 times increase absorption of a protein called casein from bodily fluids. Again, casein is known to reduce bacteria absorption. When you can look at mucin, again, another key antibacterial protein in your body, we saw 8 times increase absorption of mucin on PEKK compared to PEKK or titanium. Lubricin, of course, was invented, or not invented, but named because it is a lubricant protein in your joints. Well, it also has antibacterial properties. And we saw 3 times the increased absorption of lubricin on PEKK compared to PEKK in titanium. So clearly, PEKK has the energy, the surface energy, to increase the absorption of these key proteins known to inhibit bacteria. So of course, the last thing I want to share with you is we needed to correlate that absorption of these proteins to reduce bacteria attachment on PEKK. If these proteins are so important for reducing bacteria absorption, if we pre-coat titanium, PEKK, and PEKK with these proteins, we should see an inhibition of bacteria. So we're really correlating this increased absorption of casein, mucin, lubricin to a decrease in bacteria absorption to kind of close the circle in this mechanism. And indeed, that's what we found. So PEKK increased the absorption of casein that I showed you, which in turn, when we pre-absorbed casein onto PEKK, we saw the greatest inhibition of Staphylococcus epidermidis, looking at colony forming units. Again, looking at Pseudomonas aeruginosa, we found that that increased absorption of casein decreased Pseudomonas aeruginosa on PEKK, did not do it on PEKK or titanium. And lastly, for MRSA. MRSA, when we pre-absorbed casein on PEKK, we were able to correlate the increased absorption to decreased MRSA colonization. And I'm not going to show you all the other bacteria and all the other proteins that we looked at, but we're seeing that same mechanism as come through. In other words, PEKK has the appropriate nanoscale roughness, has the appropriate surface energy to absorb endogenously or from your body these at least 3 proteins, mucin, casein, and lubricin, which in turn are inhibiting bacteria attachment. And again, the key thing in all of this to me, and the reason why I get so excited about this material, is it's doing it without an antibiotic and not contributing to the antibiotic resistant problem that we have.